Hello learners, myself Dr. Renuka Mainde, corporate nutritionist and counselor for IGNOU in their MSc Nutrition and Dietetics course. Today we are going to learn about resistant starch. In this module, we'll be learning what is resistant starch, how to identify the calorific value of resistant starch. Module also explores the role of challenges in product development with resistant starch. Now let us understand, learners, what is resistant starch? Well, as the terminology suggests, resistant starch is that portion of starch that escapes the digestive enzyme in small intestine, which is resistant to digestion, passes through further colon. The term resistant starch was coined and defined by the group of scientists and not defined by government agencies. Resistant starch is defined as total amount of starch and the products of starch that resist digestion in small intestine of healthy people. Resistant starch that reaches the large intestine can act as a substrate for microbial fermentation, the end product being a hydrogen, carbon dioxide, methane and short chain fatty acids. Let us learn the structure of resistant starch. The resistance of starch to digestion is inclined by the nature of relationship between the starch polymer with higher amylose level in the starch being related with the slower digestibility rate. Retrograded starches states to a certain structure form of resistant starch. Let us now see retrogradation happens when the starch is cooked in the water beyond its gelatinization temperature and then cool. The starch granules gelatinize and the starch is subsequently more easily digested. In wide-ranging starches rich in amylose are naturally more resistant to digestion and also more susceptible to retrogradation. It occurs naturally in many cereal grains, fruits, vegetable, or it can also be formed during the processing of food. Resistant starch can increase the fiber intake without compromising taste, texture, and it can be incorporated into the diet. Let us now understand the different 4 types of resistant starch which is commonly found. They are labeled as RS1. This type of starch granules are physically enclosed within the cell walls of plants, hence not accessible for digestive enzymes. They are mostly found in seeds, legumes, unprocessed whole grains. The second type is called RS2, which resists digestion because of its crystalline structure. This structure is ungelatinized. This is found in uncooked raw potato, green banana, legumes like peas, beans, and cooking decreases the amount of RS2 resistant starch. Third type is RS3. This starch are formed during processing when the starch containing food are cooked and cooled. For the example, like extruded noodles, pasta, rice, potato. It resists digestion because after cooking, the starch molecules associate together again and form a compact structure. Generally, amylase retrogradation contributes more to RS3 content of the food. Let us now see the starches are chemically modified to decrease their digestibility. RS4 is a type which is found in food product for imparting desired properties such as color, stability, altering viscosity to the baked goods. RS4 is not naturally found in the food. Let us now see the calorific value and dietary intake of resistant starch. Experimentally, the energy value of resistant starch has been calculated as approximately 2 kilocalorie per gram. This is considerably lower than the energy value of completely digestible starch. Different studies have attempted to quantify the population's dietary intake of resistant starch. Most of the studies have reported that intake was ranged between less than 12 grams to 20 grams per day. Worldwide, dietary fiber intake of resistant starch are supposed to differ substantially. Dietary intakes in China and India were recently estimated at 10 and 18 grams per day. The main source of resistant starch for this cohort study were cereal, which was 42%, vegetable, which was 26%, and fruit juices, which was 22%. And the study was reported by AP Nugent. Let us now see, the learners, the measurement of resistant starch. The main step of any method to measure the content of resistant starch in the food must first remove all the digestible starch from the product using thermostable alpha amylase. It is based on principle of enzymatic digestion and measures the portion of starch resistant to digestion at 37 degrees centigrade that are typically not quantified due to gelatinization as 100 degrees centigrade followed by digestion at 60 degrees centigrade.